Hello everyone. I welcome you all once again to my series of lecture that is Understanding Pharmaceutical Science with Dr. A.J. Hariharan. So today we are going to discuss about the one of the isolation of pure culture technique, that is pour plate technique. So in this lecture, first we will understand about what is meant by the isolation of pure culture, and we will elaborately understand the steps involved of the isolation of pure culture using the pour plate technique. So first let us understand about the mixed culture. So what happens naturally is that in a natural habitat, generally the microorganism lives together. So it grows in this complex environment and we can find a mixed number of species if we take a sample from a natural habitat. So if I take a sample from a soil or from a sewage or from any water resources or air sample, in that it carries different types of microorganism, bacteria, fungi, algae, or different species of bacteria it will be present. So this we consider as a mixed culture. So in a mixed culture it is very difficult to study a particular type of microorganism. So that's why our need is that if you want to study a particular microorganism, the first step is we have to isolate a pure culture of this microorganism from this mixed environment. So that becomes the first stage. So this development of the pure culture was been put forward by the German bacteriologist Robert Koch. So Robert Koch has developed this pure culture technique because it is essential for its research purpose. That is, in its Koch postulates, he told that every infection is caused by an every infection or every disease is particularly caused by a particular microorganism. So to prove this, first he has to isolate that organism in a pure culture state. So that's why this pure culture isolation of pure culture concept was developed by Robert Koch. So first we understand about the pure culture. So pure culture is nothing but a particular population of cell is arising from a division of a single cell. If a single cell divides and divides, it forms a population of cell in the form of a colony. That is considered as a pure culture. So this pure culture is essentially needed for studying or characterization of a particular microorganism. So to study its morphological, biochemical or genetic things, we need the organism in pure culture state. So how we can get this pure culture? What is the principle behind this isolation of this pure culture? If you see that what actually has been done is that the mixed culture has been serially diluted until a relatively very low density of population of microorganism is produced. Then it is spreaded evenly on an agar surface to an individualized cells, and this individual cells has been divided and forms an individual separate colonies. This can be easily viewed with the help of a without an help of a microscope. That is, we can view this colony naturally with the help of an eye itself, and this is subculture to get the pure cultured organism. So this was the basic idea of the isolation of the pure culture. There are different methods are available for the isolation of pure culture. If you see the various methods of isolation of pure culture, the most common is the three types. One is the pour plate technique, second is the spread plate technique, and third is the streak plate technique. Out of this, today we will understand about the pour plate technique. So the pour plate technique is the very easiest way of performing the isolation of pure culture, but still it has some critical stages. So how this can be done? The first step, if you see that how the pour plate technique is done, is that first the sample is serially diluted until the microbial population is drastically reduced. Then it is plated on an agar surface. So once it is plated with an agar surface, we can get an individual colonies at an entrapped cells. So this I can show in the picture. You can see that. So the original sample is diluted serially by a tenfold dilution using a distilled water. Then we can plate at any dilution. So the appropriate dilution is identified based on the plating. For example, if I dilute at 10 to the power minus 3 dilution, we will get an overcrowded colonies. So it is very difficult to isolate the individual colonies for the study. So we can go for the next dilution. If you see to the 10 to the power minus 4 dilution, we can get an individual colony. So this can be isolated and incubated. So this is the basic principle behind the pore plate technique, how it can be performed. So if you see the stages, how it is done is that in the first step, a serial dilution is done to the sample. So we will take the sample and we can dilute serially by a 10-fold dilution, a 20-fold dilution, a 5-fold dilution. It is nothing but how many times we are diluting on each successive stages. So first we will take the original sample. So this is an example of a 10-fold dilution. So we take a sterile test tube. To this 9 ml of sterile distilled water is added and 1 ml of the sample is added. Then it is mixed well. So in this you can see the sample, how much it is diluted is 10 times to the original sample. So in the second stage, once again what we are doing is we are taking another test tube with a 9 ml of sterile distilled water and 1 ml of previously diluted sample is added. So in this stage what happened is the previous, compared to previous dilution, it is 10 times diluted. Compared to original dilution, it is 100 times diluted. So it has become 10 to the power minus 2 dilution. Then it is sequentially done. Once again from 10 power minus 2 dilution, 1 ml is transferred to another 9 ml. We get 10 to the minus 3 dilution. Then similarly 10 to the minus 4, minus 5, like that we can dilute it. So let us consider we have diluted up to 10 to the power minus 4 dilution. And the second step is mixing this diluted sample with a liquid agar. A liquid nutrient agar is prepared, then it is sterilized. Once the temperature of the medium is around 45 degrees Celsius, which is the very critical part of this pore plate technique. So at this 45 degrees Celsius, the agar will be in liquid state. Above this temperature, it will be in liquid state. Once it go below 45 degree, it started getting solidified. So at this temperature around 45 degree, a small volume of this diluted sample should be added and mixed well. So if you add more than 45 degree, what happened is the microorganism will die. If you add after 45 degree, what happened, even mixing will be not be there because the agar gets started getting solidified. So this is very important that we have to add exactly at 45 degrees Celsius approximately. So at this temperature, generally the microorganism won't die and it is evenly distributed also. So this brief exposure does not kill the bacteria or fungi or any type of microorganism in this case. Then in the third stage, after mixing in the conical flask in an aseptic manner in a laminar flow hood, it is poured into a sterile petri dishes and it is allowed to cool to the room temperature. During this time, what happened is that once the agar gets solidified, the each individual cells will be fixed or entrapped in the agar at a particular location. So after incubation, it can be grown as an individual colony. So then in step 4, the incubation is done the pet plate is inverted and kept at an appropriate temperature and duration for incubation. So after the incubation, it will be grown into individual colonies. Then finally in step 5, it is subculture. These individual colonies is added to a fresh medium and kept as a pure culture form. Then it is used for the further studies. So I think this lecture will be very helpful for you understanding about how the pore plate technique has been performed. So thank you very much. So continuously listen my future lectures in terms of microbiology as well as biotechnology. So